I will start talking about the topic at hand. You have had an excellent, unfortunately I was not there, a talk about basic research. I have been engaged in basic research for almost, from God knows, from 1972. And I want to present to you the concept, the idea that basic research is the engine that advances clinical medicine. Basic research is not something far out there, somewhere else somebody is doing it and you don't have to know about it. Basic research is the engine which brings advances in clinical medicine and practice of medicine. And I will tell you this from a personal story, from my own basic research to the clinical application. And that is my main goal, is to show you, to demonstrate to you how basic research can make improvement in human health. And I will use that, I will use my own research story as an example of that. So with that, Dr. Bedi, can you go to the next slide? So I am deeply grateful to two of my gurus. On the left side is Dr. Ramalinga Swami, and that the black and white photo. And on the right side is Dr. M.C. Dev. And these two gurus, from these two gurus, I imbibed and soaked the love for science. And I also learned from them never to say anything was impossible. Because these two people did things at a time when people thought they were impossible. And so by watching them, I learned never to think something is impossible. And I'm very grateful to my teachers. And if Dr. Dev was there physically, I would have gone and at this point bowed to him and touched his feet. But unfortunately, I can do it only in imagination and not physically, because we are not there. So the theme that I want to present to you is from bench to bedside and basic to clinical. And here is what I'm going to talk to you. I will tell you about my bench investigations. Bench means working on a research bench. And the bench part of my research was into the basic cellular and molecular biology of natural killer cells in mice, and studying the 2B4 CD244 receptor. This basic research on the bench led in an unexpected way to the bedside by my discovery of the molecular basis of a fatal human disease and the development of a diagnostic test for the disorder. I will show you, I hope to show you the connection between bench, which is the basic research, bedside, which is the clinical application of basic research. So that I want to emphasize upon you that basic research is not something which happens somewhere else. It is not something that happens 1,000 miles away or 10,000 miles away in somebody else's life. Basic research can be in your life. And you can make discoveries that will help not just our own countrymen in India, but also humankind in general. So that is my message to you. Important is this, that pay attention to basic research. Dr. Dev will fully agree that basic research is really an engine which sort of makes advances in the practice of medicine. And I hope to give you an example of that. Next slide. So as in the introduction was mentioned, my journey begins in 1974. And I hope you see on the screen a paper published in 1974. There is no need to go through the title of this paper, but this is to tell you that this was my very first and very important paper in which we made the discovery of a new type of lymphocyte called natural killer cells. And ever since that time, since 1974 to the present time, all my entire research life has been spent studying the cellular and molecular biology of these cells. A portion of that I will discuss with you today. Next slide. So for those who may have not yet who might be in the first professional and may not have had any exposure to immunology, I will just give you the basic definition of natural killer cells so that we all are on the same plane. So natural killer cells are a distinct population of lymphocytes that can kill tumor cells, virus-infected cells, and some normal cells without any prior sensitization. That's very important. T cells and B cells require prior sensitization. NK cells do not require sensitization. They are ready to act. They are like fully armed army ready at the battlefront, ready to attack. Unlike T cells, they have no memory. You know, the whole basis of vaccination is memory. That the T cells and B cells have memory. You expose them once, and when you immunize them, when the next time you get the same infection, because of the memory, they come back and quickly try to eliminate the infection. NK cells have no memory. And NK cells can secrete a lot of soluble factors, including interferon gamma, TNF, GM-CSF, and various others. And through these secreted molecules, NK cells influence the function of other immune cells, like T cells, B cells, and macrophages. So these are called killer cells, but they have more functions than just beyond killer cells. Okay, next slide. So I hope you can see on the screen a photomicrograph. Shardul, are we seeing the photomicrograph? Okay. I hope that what you're seeing is a photomicrograph, and I'm showing this to you. Has the photograph come?
Yes, sir.
Okay. All right. Shardul, anytime I say, "Can you see this?" and if you don't, just let me know. I will pause for a second. Okay.
Yes, sir, sure. Yeah.
Okay. So here you see some very large cells, which are cancer cells, and 3 small cells attached to them. 2 of them are attached to the one big cell in the middle and 1 on the left side. These are natural killer cells. They have appearance of somewhat large lymphocytes, and if the screen resolution allows, you will see they also have granules in their cytoplasm. And that's why sometimes they are called large granular lymphocytes, LGLs. Okay? This is the first photomicrograph ever somebody took of highly purified natural killer cells obtained from a mouse. And this was a very, very important step for us to go forward to work on these cells, because we had to get them in very, very pure form. Next slide. Okay, so the next slide will be have an animation, and there will be 3 panels in it. The first one, we can go to all the 3 panels. So here, so now on the top, the title of this slide is this: self non-self discrimination. Self non-self discrimination. That is, being able to tell in your body what is yours and what came from outside, what is foreign, is a very, very important property of the immune system. Otherwise, our lymphocytes will be killing our own cells, which can happen when somebody has an autoimmune disease. So how do NK cells tell which is self and normal and which is not self and must be killed? And this is explained by what is called the missing self hypothesis. What this hypothesis says is the following: each NK cell, shown in the blue on the left, has 2 classes of receptors, an inhibitory receptor and an activating receptor. The inhibitory receptor is normally dominant. An inhibitory receptor recognizes normal self MHC class 1 molecules. These are histocompatibility molecules, or antigen. Every cell in our body has it, and everybody has a unique pattern of it. So your NK cells can recognize your MHC molecules. The inhibitory receptor sends an off signal to the NK cell, so there is no lysis. When a cell becomes abnormal, for example, by virus infection or by tumor transformation, that recognition of self is abolished, because the expression of MHC molecules is reduced or absent. Now there is no off signal, and the activating receptor kills the cell. As a corollary of that, if I take your NK cells and show them my normal cells, what will they do? Well, for them, my normal cells have my MHC class I molecules, not yours. Your NK cells only know your MHC class I molecules. So they will find absence. They will find your molecules missing. That's why it's called missing self. And because they will find them missing, they will kill. So this is a basic mechanism by which NK cells recognize abnormal cells and cells which come from another individual, which of course creates other problems when one does transplantation and so on. So this is the missing self hypothesis, and this is the fundamental way by which NK cells are organized. Next slide. Okay, so as mentioned, NK cells have two classes of receptors, inhibitory and activating, and we will not go into all the details of each one of those. My laboratory worked on many of these molecules. We identified and cloned many of these molecules. But today I'm going to focus my talk on one particular molecule which you see in yellow under inhibitory as well as under activating. There's only one molecule here which is under both of those, and it is called 2B4 or CD244, and our talk will focus on CD244, which will take us from the bench to the bedside. Next slide, please. Okay, so I want you to pay attention to this slide very carefully, because this is a slide which is a roadmap. It's a roadmap to the presentation that I'm going to make to you. And the roadmap is as follows. What I have shown in the form of a sketch here is the basic organization of any cytotoxic lymphocyte. Okay, what must it have? It must have a receptor. In this case, it's shown in blue, sticking out from the surface of the cell. It must have a cytoplasmic portion through the membrane, and the cytoplasmic portion must have certain amino acids which upon activation get phosphorylated, and upon phosphorylation these sequences recruit molecules which then have to do their function of the cell. So in this case, the recruited molecules give rise to activation signals. The activation signals, which are many and not shown here, go to the granules, and they cause the granules to be injected into the tumor cells that they are attached to. Remember, I showed you the tumor cells attached to an NK cell. So if a tumor cell was attached to this cell below, those little dots that you see coming out on the underside, those are perforating granules, and these granules drill holes into the tumor cell and transfer other granzymes and other enzymes which eventually kill the tumor cell. So the entire wiring I want you to understand the entire wiring of this system. The wiring of the system starts with a receptor and the cytoplasmic portion of the receptor, which can recruit activation molecules, activation signals, and the delivery of granules into the target cell that has to be killed. If you keep this picture in mind, you will come back to it again and again, and I will show you how we move from basic science to clinical medicine. So I will start with the basics of the molecule called 2B4, which is a receptor on the surface of natural killer cells. Next slide, please. Next slide. Okay. So how did we discover 2B4 a molecule, which is a receptor on NK cells? Well, we first purified natural killer cells by flow cytometric sorting and very, very extensive sorting. I showed you the picture of the purified cells earlier. Then we took these purified natural killer cells and immunized rats with them. to create antibodies against the mouse's NK cells. Okay. Once we made the antibodies, we made hybridomers of those. We made monoclonal antibodies, and then we asked the question: what effects do these antibodies have on the function of NK cells? Okay. And that was the starting point to decide further which particular antibodies we were going to study. Next slide. Okay. Now this is a flow cytometric profile. We did not go into any details other than to tell you that when we stained the spleen lymphocytes from mice with the anti-2b4 antibody and put a fluorescent molecule on the anti-2b4 antibody, we are able to see fluorescent cells, and those fluorescent cells are the ones which have bound to the anti-2b4 antibody. So on the left side, you see the entire population of lymphocytes from mouse spleen, and you see there are only 0%. Only 0%.
The second slide, the slide hasn't changed yet.
Yeah. Okay. All right.
Yeah.
Oh, no. This is you've come one too far. Go back. Yes, this one. Okay.
Yes,
Can you see a slide with 2 panels? Can you see that slide with 2 panels, left and right?
Yes, sir, we can.
Okay. All right. So what I was about to say was that this is the flow cytometric profile of lymphocytes from mouse spleen stained with the anti-2b4 antibody that we had created. This is a way of finding out how many cells bear that particular receptor. And so in a normal spleen on the left, it's 0.9%, and in somewhat purified lymphocytes, you can see two. What we did here was we separated those 9% cells which were stained with the anti-2b4 from the remaining, and then we asked the question: which population of these cells is killing the tumor cells? On the left axis is the percent of tumor cells killed. On the right axis below is the number of NK cells for every single tumor cell. So that's a ratio. When it says 20 means there were 20 NK cells for one tumor cell. We mix them in those ratios. Now in the middle panel, you see the killing activity of the entire unseparated mouse lymphocyte population. On the bottom side, you see the activity of the 91% of cells which did not have 2b4 on their surface. On the top, left you see that population, 9% of cells which you separated, which have all the killing activity. So you see very clearly that the killing activity of mouse splenic lymphocytes is all in the population of cells that has 2b4, and none in that is 2b4 negative. I hope this is clear. So from this we knew that every cytotoxic NK cell in the mouse lymphocytes had 2b4 on cell surface. So we then next asked the question. Next slide. Sanjay, next slide. Okay. The next slide. So if you don't see the next slide, just let me know. Okay. So in this we said, okay, we have an antibody which is against some cell surface molecule on mouse NK cells. What does it do? How do we know that the molecule being recognized with this antibody has any functional effect on the NK cell? So what we did was, in an experiment which I won't go into great details, what we did was we treated the NK cells with the antibody and looked for any functional effects. And the left side, if you see 3 bars, irrelevant antibody, anti-NK1 point antibody, and 2b4 antibody. And what is shown here is the release of those granules that I showed you earlier. You notice that when the cell was triggered by 2B4 antibody, granules were released. And on the right side, doing the same thing, interferon gamma was measured. And you'll see in the middle panel, interferon gamma is released when NK cells are treated with its antibody 2B4. So we saw two functional effects of this antibody. So, which meant that whatever antigen they are recognizing on the surface of NK cells has the ability to send a signal inside the cells to secrete granules or to secrete cytokines. And this was important for us to know because we wanted to work with those antibodies that recognized function-associated molecules with the NK cells, of the NK cells. So having satisfied ourselves that the anti-2B4 antibody recognized an important molecule on the cell surface which could signal the cell, we went on to the next step. Next slide, please. So, it was what we knew it had effects. Once we knew it had functional effects, we cloned the molecule, and this was done by a very talented young Indian scientist who came to me from National Chemical Laboratory in Pune. His name was Matthew, and Matthew cloned 2B4, and in this picture he shows we see that this particular molecule in diagrammatic sketch 2B4 is a part of a family of molecules which have more or less similar organization. This family of molecules is called the CD2 family of molecules, because CD2 is a founding member of this particular family, and everything else was discovered afterwards, so therefore it is called the CD2 family. I won't go into molecular details, but they have in general a common structure in which they have an outer portion with 1 or 2 loops, or 3 loops or 4 loops sometimes, in a transmembrane portion, and they're sort of otherwise there is a lot of homology, similarity at the amino acid level between these. They are not the same, but they belong to the same family. Just like, you can look at your cousin and somebody says, "Oh, ye aapka bhai lagta hai." They can tell that because there are certain genes you share, and therefore you have certain common appearances. So at the molecular level, the same thing happens. Ye sare bhai behan hai in this family. But our interest is in 2B4. So next slide, please. Sanjay, next slide. Okay. Now, what we saw when we did a careful amino acid analysis of the cytoplasmic portion of the 2B4 receptor, we found a very interesting thing. We found amino acid sequences of a certain kind. And again, it's not necessary to go to the sequences. There were 4 set of sequences in the cytoplasmic portion of 2B4, which together formed a unique sequence, and we'll soon talk about what the significance of this sequence is. Okay, this sequence was called immunotyrosine switch motif, ITSM, as you can see in the title. Okay, just keep this in mind. Next slide, please. We subsequently learned that the 4 members of the sequence family. We subsequently learned that 4 members of this family share these immunotyrosine switch motifs in the cytoplasmic portion. They are all shown here on the left side, and so this broke the family into 2 subfamilies: those which have ITSM, those which don't have ITSM. And the importance of ITSM is that ITSMs, this sequence of amino acids, when phosphorylated, can attract other proteins from the cell, and one of them written here is called SAP. And so it's a small protein recruited by the cytoplasmic portion of these receptors when these cells are activated, and this SAP molecule can then attract other types of molecules which can either activate the cells or shut off the cells. Okay? Just remember at this point 2 things. There is something called ITSM in the cytoplasmic portion, and to it can bind a small protein called SAP. Okay? Keep these 2 things in mind. We will come back to them later. Next slide, please. Okay, then we had to find, we knew that 2B4 was a receptor. We have to find what its ligand was. What does it bind to? I mean, all receptors have ligands. Insulin receptor has insulin as a ligand. Growth factor receptors have growth factors as ligands. So all receptors have some ligands, which are proteins that bind to them, and when they bind them, they activate them. So the ligand for 2B4 is a molecule called CD48, which was in the same family actually. And we didn't do this work completely by ourselves. It was done with the help of people in Oxford in England who were experts on the C2 family. And in a collaboration, they helped us identify CD48 as the high affinity ligand for 2B4. Now CD4 is expressed on all hematopoietic cells, not just NK cells. Every hematopoietic cell, T cells, B cells, NK cells, dendritic cells. It was also known earlier that when a cell is infected with a virus, the expression of CD48 goes up. This will also become important later on. And as I said, we learned that 2B4 and CD48 can bind each other. Next slide, please. Sanjay, next slide. Okay. So here is the summary of all that I have told you so far. 2B4 is a receptor of the CD2 family that is expressed on all NK cells, memory CD8 T cells, and all gamma delta T cells. CD48 is the ligand for 2B4, and its expression is on all hematopoietic cells. Cross-linking of 2B4 by its ligand or anti-2B4 antibody activates NK cells. I showed you the activation thing, that more interferon gamma was coming out and more granules were coming out. So you can either, the 2B4 molecule is a receptor sitting on NK cells. You can trigger it either by putting the antibody on it, which then connects receptors which are in the close vicinity by cross-linking them, or you can do the same thing by exposing it to a cell that expresses CD48, which is its natural ligand. Okay, so either way, this molecule can be triggered. Next slide, please. Okay, so we come back to the roadmap. I just told you about the 2B4 molecule, and on the left side you see we'll in red we will now move to the next part of the story, the ITSM motif, the SAP molecule, and XLP. And XLP is a human disease, and so we'll see how we change from study of mice to study of humans. Next slide, please. Okay, this is a brief description of this human disease called X-linked lymphoproliferative disease, and in abbreviation XLP. Now, many of the seniors in the audience will know very well that this is an immune deficiency disease, in which the disease occurs when the person is affected by Epstein-Barr virus. Most of you know that Epstein-Barr virus infects all of us, and we don't even know about it because our NK cells and T cells can just take care of it. In some people, it gives rise to infectious mononucleosis, and in most healthy people, that also gets cured by itself. But in certain individuals, there is a genetic deficiency in the ability to control Epstein-Barr virus infection. And everybody has Epstein-Barr virus, so everybody is exposed to it. In these individuals, 85% of the people, of the boys, obviously it's X-linked, so it's a males only, 85% of the people die, compared to no effect on you and me. Okay? And of the people who die, about half of them die with a disease called fatal infectious mononucleosis, and this is a specific syndrome in which there is a massive proliferation of T cells and macrophages, and the person actually dies not from the virus but from the overactivity of the immune system. And we'll come back to that. And other 50% die by different ways. Now, the reason I'm bringing this to your attention is this, that while we were doing this research, another researcher in Boston discovered that the gene that was mutated in patients who had XLP was the SAP gene for the SAP protein. And I just mentioned to you earlier that this SAP protein can bind to the cytoplasmic portion of 2B4. So we now have a connection between the cytoplasmic portion of a mouse receptor and its counterpart in humans and its association with a human disease. So you see we are gradually moving from the bench to the bedside, from basic to clinical. Next slide. Okay, so just briefly, what is SAP? As I said, SAP was discovered by its interaction with the cytoplasmic portion of one of those molecules, CD150, a member of the CD2 family. It's a small protein. It can bind at special tyrosine-containing motifs called ITSM. I've showed you those. It's expressed on T cells and some B cells. And when there's a mutation of this particular protein, the boys get X-linked lymphoproliferative syndrome. So there is something about XLP that makes people become susceptible to infection by Epstein-Barr virus leading to their death. And this has remained a puzzle and a mystery for a very, very long time. Next slide. So coming back to this roadmap slide, we talked about 2B4. We talked about the amino acid sequences, and then we talked about the recruitment of this protein called SAP to those sequences. And I just told you that around that time a discovery was made that if the SAP protein is mutated, the result is the human disease XLP. Now we are firmly moving away from the mouse and moving to the human, because now we have become very, very curious as to why and how this XLP disease is caused in human, a uniformly fatal disease. So our attention is now focused on XLP and XLP-like diseases. So the next slide will show us the group of diseases which resembles XLP. Next slide. Oh, no, we first asked the question, is XLP caused because of a defect in signaling function of 2B4? The reason we thought of that was, first of all, SAP mutation causes XLP. Secondly, 2B4 can bind to a SAP. I told you those four motifs. SAP binding is essential for 2B4 mediated activation, which I didn't show you. And the cells infected by EBV are B cells, and they express CD48. And when these B cells are infected with EBV, the CD48 level goes up tremendously. So for all these reasons, we first speculated that 2B4 defects may be involved in the causation of this human disease XLP. Next slide. Okay. I can see the slide, so I think you can see it also. So we made a little cartoon to show, to ask the question, is 2B4 involved in human disease XLP? And you see in the cartoon here, on the left side, a killer cell, on the right side, an EBV-infected B cell. The EBV-infected B cell has CD48 molecules on its surface, which increase after EBV infection. Okay, that is why it says CD4+++. And we know that the 2B4 receptor binds to CD48. So the hypothesis was that because there's a mutation in SAP, this interaction between 2B4 on the NK cell and CD48 on the EBV-infected cell fails to send the signals. You see an interruption in that arrow there. Fails to send those signals which are required for the cytotoxic function to take place. And therefore, the EBV-infected B cells keep on proliferating and the other cells keep on proliferating and the person dies. So the question mark for us was defective 2b4 signaling, question mark, does it cause XLP? The next slide showed, next slide, Sanjay. The next slide showed us, oh no, we are not sure because SAP can bind to ITSM on 2b4, on CD150, on CD84, and LY9. Because all these four molecules contain the same amino acid sequences. So if a mutation in SAP protein causes XLP, it could be because of any one of those four receptors, functioning of any one of those four receptors, not just 2b4. So this is called correlation. So what we are looking at was correlation. Correlation is not causation. So when there's a correlation, then you have to ask the question, is this just a correlation or is it also a causation? So when we saw some reasons for it might be involved, but we saw it as a correlation, and we couldn't be sure that this correlation was because of defect in 2b4 function resulting from mutation in SAP. So we decided that the only way we will find out without any doubt that 2b4 is involved in causation of a human disease would be to look for human beings who have a genetic defect in 2b4. Next slide. So the title says Basic Science to Bedside, Changing of the Hat. What I mean by changing the hat is this. So far I was wearing the hat of a basic scientist. Now I change my hat to be a hat of a clinically trained person and a pathologist. And this is again the intersection of basic sciences with the practice of medicine. So we ask the question, which is on the right side there, is there another XLP-like syndrome in which the genetic defect is 2b4? So you notice that now we have moved firmly away from the mouse NK cell to a human disease. And now we are very curious whether defects in 2b4 can cause a human disease. And so we started the search, we changed the direction in the lab, and we started the search for diseases which resemble XLP. We knew XLP is caused by SAP mutation, and we thought that anything that resembles this XLP is likely to be due to genetic defects somewhere along the pathway of 2b4 receptor and below that. So that was our starting hypothesis. Next. So we started searching the literature and tried to find as many diseases as we could which had been described previously with unknown cause, okay, which had some similarity with the XLP. Okay. We were looking for something like XLP because we thought we will find something which has a mutation in the 2b4 gene. That was our purpose. So when we looked at other diseases which resemble XLP, we found there are many, and they are together called HLH syndromes. Some of them are inherited, like XLP, like a disease called FHL, and other two diseases, Cherekiahashi and Griscelli syndrome, and a much larger number of acquired syndromes, which are called virus-associated hemophagocytic syndromes. And in these individuals, after a transplant or a leukemia or lymphoma, they would get virus infections and get a picture which just look like XLP, okay, even though they didn't have a genetic defect in that in SAP. So we started searching for those. So in our search, we were looking for candidate human diseases that would be caused by our gene of interest 2b4. Next slide, please. Okay, so we became very interested in this rare disease called FHL, familial hemophagocytic lymphohistiocytosis. And it is an autosomal recessive. It affects 1 in 50,000 live births. And these infants below the age of 2 years get a trivial virus infection and they die. and without bone marrow transplants, they all die. So, and not EBV virus, other viruses, any kind of virus infections would kill them, whereas a normal healthy infant would not even know that he or she had an infection. And so this clearly is a genetic defect, and it was inherited and autosomal recessive, and its clinical picture was not very different from the XLP, which is what we were looking for. Okay. Next slide, please. Sanjay. So just to give you a little clinical picture of FHL, these small children present with fever, enlargement of liver and spleen, hepatosplenomegaly, pancytopenia, because the enlarged spleen starts eating up the red cells and white blood cells and removing them from circulation. And very characteristics, there's an accumulation of hyperactivated macrophages with massive proliferation of and activation of CD8 T cells. And these two cells, macrophages and CD8 T cells, produce gallons and gallons of cytokines, tumor necrosis factor, interferon gamma. And the children die not from the virus, they die from the overactive flood of TNF and interferon gamma called cytokine storm. So we, as I mentioned, we got very interested in this because it resembled XLP. We were looking for a disease that resembled XLP to look for our human being with gene defects in 2b4. Next slide, please. Okay, so the important part of this slide is the statement at the bottom. Here, boss, that is me, says to my PhD student, step, Susan, is 2b4 involved in the pathogenesis of FHL? Have we found a human disease with a mutation in 2b4? Let us find out more about this thing. Let us try to test this hypothesis. Okay. Next slide, please. We maybe skipped one slide, but that's all right. We can talk about this. It's okay. It's okay. So while Susan Stepp, my PhD student, and I were speculating that the disease FHL may be caused by a mutation in human 2b4, a paper came out. The paper came out and Susan came running to me and said, boss, our hypothesis is incorrect. And I said, why, Susan? Why is it incorrect? She said, well, look at this paper just published from a very distinguished French group of scientists who worked in Institut Pasteur. These people are experts in human immune deficiencies. They study FHL. They have followed up families. And they just published this paper in American Journal of Human Genetics in which they did a linkage analysis of this disease called FHL. Linkage analysis is a method to link the causation of a disease with a particular part of a chromosome. That's what linkage analysis means. You link the disease to a part of the chromosome. And there are various ways of doing it, but these people did linkage analysis, and you can see the title. It says, Linkage of Familial Hemophagocytic Lymphocytosis to Chromosome 10q2122 and Evidence for Heterogeneity. So in this paper they said 2/3 of the patients who have this disease FHL, is linked to chromosome 10q2122, and the rest are linked to one or two other different parts of the genome, and this is a heterogeneous disease. So why did Susan say this hypothesis is incorrect? Let's look at the next slide. So this is the map of human chromosome 10 on the left, human chromosome 1 on the right. So according to the French paper, the disease FHL had been linked to a portion of human chromosome 10 called 10q22. Okay, these 10q22 these are just ways of naming the regions of the chromosome. Okay, chromosome 10, Q means long arm, P means short arm, and then the Q long arm is divided into certain segments, and this one was a 10q22. Now the reason why the hypothesis was wrong was because our gene 2b4 we knew mapped on chromosome 1, not on chromosome 10. We had already mapped it. It mapped on chromosome 1, 1q, long arm of chromosome 1, band 23. So the power of genetics was telling us that FHL, which maps on chromosome 10, and 2b4 which maps on chromosome 1, it cannot be that a defect in 2b4 gene can give rise to FHL. They are on parts of the genome. So we didn't stop there. Next slide. We asked ourselves. I'm waiting for the next slide. Yes. So in the red color on the below, myself, asked Susan, what else maps to 10q22? And we put a question mark there. And the answer, next slide. The answer, you see the answer there. The answer is perforin maps on 10q22. So they had shown that the FHL disease maps to 10q22. We showed right next to it is perforin gene sitting there. And we said, aha, we think we know the answer. Next slide. So coming back to this particular roadmap, we talked about 2b4 receptor, we talked about SAP, we talked about SAP mutation causing XLP, and now we were looking for the cause of FHL. And whereas we were looking for the cause of FHL as possibly the 2b4 receptor, what the evidence was telling us was, no, it was not the receptor. Okay, it was something which was downstream from the activation signals, and the last part of the activation signal is the perforin protein, because that's the one that leaves the cell to kill the tumor cell. So we were looking for the defect on the proximal side. The data was showing us it has to be on the distal side. Just remember, any mutation in this pathway from 2b4 all the way down to the cytotoxic granules is going to give rise to exactly identical looking disease, because the same pathway that has been affected. And so this then gave us the hypothesis shown on the next slide. So we wrote down our hypothesis that the primary inherited defect in 10q22 linked FHL is impaired perforin-mediated cytolytic function. Okay. So having made that hypothesis, how did we test it? Well, I picked up the phone and called Paris. And I said to Dr. Saint-Basile, who was the senior author of the paper, Dr. DeBasile, we know what causes FHL. Send us 8 samples of DNA, 4 from patients and 4 from controls, and label them A, B, C, D, E, F, G. Don't tell us which is what. And within 2 weeks, we'll tell you which DNA samples were from patients and which were from controls. She said, but how? What are you going to look for? We said, we have a hypothesis. Okay, we won't tell you the hypothesis until we have tested it. You can send us 8 DNA samples, unless you break the code, we will know nothing. So she said, fine. So I remember very well, it was May 29th, and on 2 days later, these samples arrived, a Federal Express envelope arrived. DNA is very stable. We found 8 vials, numbered A, B, C, D, E, F, G, H. We, in the meantime, had prepared primers, PCR primers, for sequencing the perforin gene. Next slide. And so we sequenced the perforin gene. And as we had suspected, we found that 4 samples had mutations in the perforin genes and 4 samples did not. So we knew exactly which 4 samples came from patients. We knew exactly which 4 samples came from normal people. And I won't go into the sequencing reactions. They're actually very simple to do. Unfortunately, perforin gene has only 2 coding exons, exon 2 and 3. We completed this entire project within 1 week after getting the samples. And we pinpointed the mutations in perforin gene. And after having done that, I picked up the phone and I said, Dr. T. Saint-Basile, these are the patients, these are the controls. She said, you're absolutely correct. How did you find out? I said, we found out because we thought that the defect is in the perforin gene. We sequenced the perforin gene. And she was surprised because they had also the same data that we had when they did the linkage analysis. They also knew there are 10q22 perforin gene maps. But as they say, luck only favors the prepared mind. And so you say we were lucky, but we are lucky only because we were prepared mentally to take advantage of the information as it became available. So after that, they sent us more samples, and you see at the bottom, you see all these PISA patients, patient 4, patient 29, patient 11, 25, patient 5, 11, patient 6, patient 34, and patient 5. So 1, 2, 3, 4, 5, 6, 7, about 12 patients they sent us later on. Every single one of them had a mutation which caused a stop codon or blocked the synthesis of the perforin protein, and none of the controls had it. So now we had genetic evidence, okay, that FHL is caused by mutation in the perforin gene. We wanted functional evidence and morphologic evidence. We did a couple of additional experiments. Next slide. So in this, so let me remind you that these children who have FHL die very young. So there were all these DNA samples from patients who had already died. They just had been kept by the investigators. They had also frozen their lymphocytes. And so we asked them to thaw their lymphocytes and test their ability to kill tumor targets. And if the hypothesis that perforin gene mutations are the cause of disease, then we would predict that the lymphocytes obtained from patients, P11, P5, P25, P21, would be very poor in killing activity compared to the controls. And you can see exactly the same thing happened. On panel A, the control killing goes up to 75%. In panel B, it goes up to about 75% too. But the patient sample 25, 21, 11, and 5 in below 20%. Marked reduction in the killing activity of the lymphocytes from these patients. Next slide, please. The next slide we should be for you to see, because I don't know how the lighting is in your room. Microscopy, in which one can look at the components of cell by putting appropriate dyes. The bottom line from this experiment is that we look for the presence of perforin protein in the lymphocytes of patients and controls by using a certain dye to label perforin molecules. And hopefully on the rightmost side, you can see a panel called control, and you can see in the middle panel there are green dots. Those are perforin molecules, okay, perforin protein. And then if you look on the left side, patient 25 or patient 21, you see those green dots are missing. So we now had three lines of evidence: genetic evidence, mutation in perforin genes, functional evidence that the killing activity was reduced, and morphologic evidence that the perforin protein was missing. So with these three together, we had powerful evidence that the human disease FHL is caused by mutation in the perforin gene. We now knew pretty much everything about the HLH syndrome, and we knew from other people's work that XLP is caused by SAP mutation, and our work showed that a similar disease called FHL is caused by perforin mutations, mutation in perforin genes. It's very important to keep an open mind when you are doing research. You may have a favorite hypothesis, and if that turns out to be not true, don't discard it. Find out a better hypothesis, and that you can actually go forward and prove. And this is exactly what we did. We thought we were looking for humans with 2b4 mutation. We found humans with perforin gene mutations. And and an important advance was made. And then basically what we said was a mutation anywhere distal to the receptor, where the arrows are, up to the end, can cause diseases which look like FHL. Okay. So that is the conclusion that we made. And as I said, we were working on mouse 2 before, and we ended up with a human disease. Next slide, please.
Excuse me, Dr. Vinay Kumar, sir.
Yes.
Our next speaker is in line, and can I request you?
Sure.
Next 5 minutes, please, sir.
It'll be done in 5 minutes. We are there. Okay. The conclusion was that genetic defects in perforin caused 22q-linked FHL disease. I think the second conclusion is very important. We predicted that other cases of FHL and other HLH syndromes are also caused by molecular lesions that impair the cytolytic machinery of lymphocytes. We made this prediction. We said that those other unknown genetic cases will also be found to have mutations in the exactly same pathway. Next slide. And here is 2015. In 2015, now we know there are 5 variants of FHL. The one we described is FHL2. All the others described have different defects in the cytotoxic machinery, which is exactly what we had predicted would happen. Different genes will be found, but all of them will be in that same particular pathway. Next slide. And much to our delight and surprise that in June of this year, the Journal of Immunology picked our paper, which was published in Science, as a pillar of immunology. You can read that, pillar of immunology. And there's a commentary called "Kill or Be Killed" by 2 other experts, and then there's a complete reproduction of our paper in the journal. And we found out that from the time we wrote this paper in 1999 to the present time, this paper has been cited 1,000 times by other people. It actually, the reason it was classified as a pillar in immunology is because it opened a brand new field of investigation in immunology. You know, a completely new, fundamental new concept of how host defense takes place. Next slide, please. That'll be the last. Of course, we were delighted to hear that our paper had been picked like that. So, my dear friends, this is the last slide I will show you. This is not science, but this is what I learned from science. Be fearless. Take the road less traveled. Never be afraid to go somewhere that other people have not gone, because that is where discoveries are to be made. Be very rigorous. Do not look for shortcuts. Do not look for shortcuts, because if you look for shortcuts, you'll make mistakes. Being right is far more important than being first. There's always a race. Who was first? Who was first? That's not important. What's important is who was right. And in many examples, people were first, but they were wrong. People who came later on were right. Share knowledge openly. Science is for sharing. Don't hide it. Don't be afraid to tell other people what your research is, for the fear that they will copy you. Be respectful of junior colleagues, because as you get senior, you will realize all the new ideas are coming from below. And if you're not respectful to junior colleagues, you'll miss out the new ideas that are coming from there. I got a lot of new ideas from my PhD students and medical students. So I learned that. Compete with yourself, not the others. Do not set anyone else as your benchmark, otherwise you will only be as good as they are. If all you want to be is as tall as the tree, you'll be as tall as the tree. That's all. You'll say this is the benchmark. But if you say, I want to reach the sky, you will not reach the sky, but you may reach 100 miles into the atmosphere. So don't set anybody as your benchmark. And finally, as Dr. Devan Ramlinger taught me, remove the word impossible from your dictionary, because impossible is just the limitation of your imagination. This is the most important message I want to convey to you. And if I may ask you to repeat after me, remove the word impossible. Repeat, remove the word impossible. I didn't hear anything. Thank you very much, and thank you very much all.